Decree today that life is taking, I'm not living, it's mine, and it owes me living. But ask me why, and I'll spit in your eyes. Oh, ask me why, and I'll spit in your eyes. But please, no dreams anymore, no. That's the body, mind, and does the mind rule the body. The iron bridge, sit up and wasn't like the old days anymore. The wind wasn't like the old days.
It's called.
You sound like you're sick. What a demo!
Heaven and hell, a great big fictitious game. Where to live is to know your shame, all your shames and your pains. To be in search for plain old happiness. Cause you are a special one, God. And let nobody put you under. Whenever your friends, they have the doubts and the words you know that just not true. To die. You just don't have me so much. It gives you love that from your heart, so a little deal. Embrace all your things. Good feeling. Rocking and a reeling. Loving and a teasing. Kissing and a breathing. Hey, good love charm. Laying in my arms.
Welcome to MetPhys 2020, Metabolic Physiology in Isolation. Or as we like to call it, CRIBS, Academic Office Edition. Metabolic Physiology in Isolation is a new model for truly inclusive global meetings. The White Megara Lab is proud to present Metabolic Physiology in Isolation free to all, anywhere across the globe. Our feelings are that it's a new model because it's both cost-effective and green. There's nobody taking any flights anymore. And it's inclusive because the talks are online, available to be listened to anytime, any place, at any moment. We've been blown away by the response on Twitter. You can use MetPhys 2020 hashtag throughout the talks to tell the speakers about what you like, ask some questions, or just share the way you're listening. Cassidy, listening at home, this is your chance to go for that Inception prize for MetPhys 2020. So far, you've got 4 days of taking photos of yourself on the screen in a row. Can we make it number 5? I think you can. I think you can get all the way out to the end of the talks. That's going to be one heck of a photo. We've got fantastic pictures of families enjoying listening to talks together. Here we have a father-son duo that has incredible smiles, enjoying the science. Jan Personas has the right idea, got this up on the TV so they can just sit back and relax and listen to the fantastic science. Dave Bridges really did an amazing job last week, sharing all the unpublished data. I think he set the bar really low for published data and really high for unpublished data. So he's going to have everyone else jumping through hoops to kind of match that. John McGavick, don't worry. If you have to make flowers or pipe cleaners like many of us do, I know I've got a few at home and many others do, don't worry about it. The talks, after they finish streaming on Facebook, are automatically listed as recordings. I'm also now uploading those recordings to YouTube for all the listeners who don't want to go to Facebook. And so they're now all on YouTube as well. And this will continue and they will stay up there without a time limit. So you can listen to them anytime you want. And multiple times if you like. Don't forget to share your MetPhys 2020 Isolation Challenge. I know we're all going crazy being locked by ourselves. Martin and I decided to try a new challenge for the period that we're isolated. You can see what we're trying to do up top. Patrick Swayze does it great. We got the first part of the move right, followed by a little bit of disaster. The chair's okay, my back's okay. We're gonna keep trying. I promise we're gonna get you a completed full-form version by the time this isolation is over. I'd love to see you all trying your own challenges. Please don't try this at home. I don't want to be responsible for anyone's bills with the doctor. But yeah, something difficult that you have to learn. Show us the fails, show us the success. We'd love to see it. Of course, there's a prize, but there's not a prize. As with everything else in MetPhys 2020, you can win, but you'll get nothing. All right. I really do want everyone to take notice of these faces. These are the people who replied really quickly to get this off the ground. They're fantastic speakers, often invited to world-class conferences, and here they are presenting their most new, exciting work to all of us, allowing it to be available freely to everyone. The way you can thank them is if you see them, tell them thank you, tweet with them during the show and after the show using that hashtag. And if you see them in person, tell them thank you. If it's at a coffee shop, buy them a coffee. If it's at a bar, buy them a beer. I'm sure they'll enjoy that. And that's what this community is all about. And as you can see, we've got lots of great talks coming. The talks that we've had already, including my own, are already on Facebook and on YouTube. It's under my name, Philip White. If you use the hashtag MetPhys2020, that'll help it show up. I think as more of us download those, it's going to be easier to find. But they're up there and they're going to keep going up there. So enjoy those and share those with your colleagues who aren't on Twitter, who aren't on Facebook. Share them with your grad students, your undergrads who want to know what real research really is. This is an opportunity for them to get to hear world-class talks, exciting research, and be inspired. I know I am. If you want to thank me personally, I wouldn't be here without the Pathway to Stop Diabetes research program from the American Diabetes Association. If you'd like to give anything at all, that can be $1, $5 to the program. In my honor, I'd be truly proud, but you don't need to do that. Just sit back, enjoy the show. The White Megara Lab is very happy to sponsor this program, to make it available for all. We're covering the cost for the Zoom room for this month so that no one else has to pay a dime. And we're really glad to do that. So after today's talk with Zach, don't forget to tune in tomorrow, where we get to hear from Rachel Perry on her new work in her own lab. And I think we're all excited to hear that. We know Rachel's been doing fantastic work for many years, really making a lot of groundbreaking strides. And I'm sure that'll be true tomorrow in her talk. And I think we're all going to enjoy that. So without further ado, I'm going to transition to Zach. I know he's ready to go. I'm going to make him the host and he'll take it away. Enjoy, everybody.
All right, hello everyone. Thanks again to Phil for getting us organized here. I had a couple visitors who wanted to start off. 3 visitors, in fact, who wanted to at least say hi. Girls, can you say hi to everybody?
Hi.
Okay, go have fun. Really awesome initiative by Phil to get this going. So I'll go ahead and get started with my talk. So my lab is interested in cancer cell metabolism and how metabolic changes can impact whether cells live or die, particularly in the context of matrix detached cells. So just a couple things right off. I am, again, my name is Zach Schafer. I'm at the University of Notre Dame in the Department of Biological Sciences. Here's a shot from our lovely campus. I've listed my Twitter handle here. I'm happy to, I'll hop on Twitter after this and happy to take talks or happy to take questions on anything, anything that comes up in the talk today or anything you want to ask about. Just use the hashtag, the MetPhys2020. Couple things just before we get started. I just want to shout out to Phil and the White Magera Lab. Awesome initiative to really to put this together. Just happy to be able to participate in it. It's I do agree with what Phil said. I think this is something that could really represent the future of scientific communication. And these are abnormal times, and it's been really refreshing to be able to sit back and just listen to some great science. And happy to have the opportunity today to present some of the stuff we've been working on over the last several years. A couple things. I know you guys saw some of my kids. We have a pretty big household, but I'll give you. I'm sure you guys are all. Everybody at home is dealing with similar things. What are the types of things you do during the COVID knockdown here? So I just put in a couple pictures before I get started. These are my oldest 2 sons. This is Clue Master Detective, which I highly recommend if anybody hasn't played it before. I used to play this probably when I was in like 10 years old myself. And so I busted that out with my 2 oldest sons. We got some good time there. You saw my 2 daughters. This is one of them doing e-learning. Here's another one who just decided to buy a robe, which we bought on Amazon. So that's kept her pretty entertained. And then my youngest guy here, he is jumping on a trampoline where he figured he could just hang on the bar himself. And what I was most proud of is my son Ethan and I came up with this game where we put army guys on the mantle in the house, and then we shot them from like a sniper's nest up here upstairs. So this is how we're spending our time this is how we're spending our time during the lockdown, and hope that everybody is staying healthy and staying safe. Okay, so I'll go ahead and get started. So my lab, we're really interested in understanding how cells survive, particularly in the context of lack of integrin-mediated attachment to extracellular matrix. And I know many of you have likely heard of these pathways, but I'll just go over them just to get us all on the same page here at the beginning. Probably the most well described pathway is the most well described change that you see when cells lack matrix attachment is this death program known as anoikis. So it's a Greek word, it means homelessness. And what you're looking at, anoikis is basically caspase dependent cell death that is a consequence of ECM or extracellular matrix detachment. So here you have a visual of cells attached to extracellular matrix. They lose that attachment and activate this program anoikis. Okay, the fact that this is caspase dependent is actually really critical because we're going to talk about today, we're going to talk about caspase independent effects that might be, that are potentially going to compromise viability for cells that lack matrix detachment. Now, when you think about this in a disease context, I think it makes quite a bit of sense that if you think about cancer progression, that cancer cells are typically going to acquire some type of resistance to anoikis. And if you think about that, I think again this is pretty intuitive, right? Cancer cells are going to travel and exist in multiple different matrix environments, and as such, the ones that actually survive and seed distant metastases, those cells have found ways to shut off death programs like anoikis. So here's a schematic that I put together with a couple of my former graduate students. We wrote this in a review article many years ago now, but we were posing a simple question here: where might anoikis inhibition be relevant in the context of the metastatic cascade? And we posited that it could be really at any stage, and even at early stages. So just to sort of give you a reason, just to sort of give you some background as to why we think that. Here, if you think about primary tumor formation, a lot of early non-malignant lesions start with cells that populate luminal space. So those are not even cancerous at that point, but yet we know that when cells tend to populate luminal space in sort of in ductal, in sort of a mammary duct or something along those lines, that potentially that those cells are going to lack matrix detachment or lack proper matrix attachment, and therefore inhibiting anoikis can be helpful for those cells and their capacity to survive. Cells in the circulation would also be in, they would not have proper matrix attachment, and therefore shutting off programs like anoikis might matter. And even in distant metastases, shown here in this diagram with the lungs, you've got cells that are in an unfamiliar matrix environment, and therefore they are they would benefit from shutting off death programs like anoikis. Now, one model system that has been really helpful for understanding how cells can survive in matrix-attached conditions is this three-dimensional culture model using MCF10As. And this is a model system that was developed by Joan Brugge. I was a postdoc in her lab where we learned to use this system. And Joan was really inspired by the work from Mina Bissel's lab, who had sort of been the pioneer in three-dimensional culture studies. So the way this system works and why it's so valuable for these questions, if you take MCF10A cells, these are a non-cancerous mammary epithelial cell line. You grow these cells in matrigel, and you start to get these spheres. Think of these as sort of a cross section of a sphere that you will, if you're looking through a cross section of the sphere. If you monitor these structures over time, you'll start to see those centrally located cells die off, and you end up with what looks like a hollow lumen. And those really do look like the ducts that you see in the mammary gland. There's a lot of characteristics of these structures from the standpoint of polarity, from the architecture that seem to suggest that this is a good model for mammary gland morphogenesis. Now, for us, what we're really interested in is this clearing process. And you can see in this diagram, there's this red line around the outside of the structure. That's representing basement membrane, which is rich in extracellular matrix proteins. Those cells in the middle, in that luminal space, don't have access to the basement membrane, and therefore they're dying by an anoikis-like death. They're dying as a consequence of matrix detachment. Now, an interesting variant of this experiment, if you overexpress an anti-apoptotic gene like BCL2, this shuts off caspase-dependent death. by blocking cytochrome C release from the mitochondria, you'll get something like this. So you do get structures that maintain luminal filling where these cells are now protected against death. But what's interesting is that that protection is temporary, and over time you lose those cells as well. Now, if you do another variant of this experiment, if you put in an oncogene like ERBB2 or HER2, overexpressed in 30%, about 30% of breast cancers, you get multi-acinar structures. So things look very different. But if you look at the actual cells in the luminal space, you now have long-term filling. So there's something about ERBB2 that gives you long-term filling in the luminal space that simply overexpressing BCL2 does not. So it's not enough to simply block anoikis or caspase-dependent death. There's some sort of other activity that's responsible for clearing these matrix-detached cells, and signaling from a receptor like ERBB2 can actually rectify whatever those problems are and permit long-term survival. And to sort of make a long story short, as to when we think about how is it that ERBB2 promotes luminal filling, it's actually intimately linked to changes in cell metabolism. And this is from work that again was part of my postdoctoral studies in Joan Brugge's lab. But one of the things we found is that in normal, non-transformed or non-transduced cells, that matrix detachment is a signal for defective glucose uptake. And it has to do with the availability of the glucose transporters on the plasma membrane. So in these normal cells, or in these non-transformed cells, glucose uptake is defective. You have limited flux through the pentose phosphate pathway, shown down here, abbreviated as PPP. That pathway is critically important for the production of NADPH. NADPH is important for neutralizing oxidative stress, and as a consequence of limited flux through the pentose phosphate pathway, you end up with elevations in reactive oxygen species. And through a pathway we don't fully understand yet, those high levels of reactive oxygen species can be toxic for those cells, and they can also interfere with ATP generation through a pathway, through a mechanism that we think depends on fatty acid metabolism. Now, if you do this with ERBB2 cells that express ERBB2, ERBB2 rescues glucose uptake, it rescues pentose phosphate flux, it rescues production of NADPH, and as a consequence, you have lower ROS, you alleviate that toxicity associated with high levels of reactive oxygen species, and therefore alleviate the dysregulation of fatty acid oxidation. So basically what this tells you, if you sort of step back, what this tells you is that if you're a matrix detached cell, that not only are you going to have to block, you need to survive, you're not only going to have to block anoikis, you're also going to have to contend with these metabolic deficiencies. The fact that matrix detachment itself is a signal for rewired uptake of glucose, those types of things are also going to impact whether those cells, matrix detached cells, are going to live or those cells are going to die. And it leaves us with a model for thinking about cancer. And we think about cancer cells that would be, during the course of tumor progression, that it's not enough to just block anoikis, that you have to block anoikis and you have to rectify metabolic deficiencies in order to survive when you don't have normal matrix attachment. So this is just a schematic of a couple of ways this can happen. But the question really that we've been thinking about a lot in my lab is, how is it that cancer cells do this? What are the strategies used by cancer cells to rewire metabolism and therefore to ameliorate some of these elevated levels of oxidative stress, and therefore to allow for survival that could contribute to tumor progression and metastasis? Okay. So I thought what I'd do today is really just talk about two projects, two stories that we've been working on quite a bit here in the lab. And the first is related to this question: how is it that matrix detached cancer cells actually die? We know that anoikis can kill these cells, and we know that that's a caspase dependent apoptotic death. But the other thing we know is that these metabolic changes actually can cause changes. These metabolic changes don't actually cause apoptosis, they cause some sort of non-apoptotic death. So the first study I'll talk about is our exploration of how these cells actually die in a non-apoptotic fashion. And then the second story I'll tell you about is how is it that signal transduction can rewire different metabolic pathways? And we've become very interested in this one kinase, SGK1, I'll get into this in a second, and how SGK1 can rewire metabolism to enable survival during matrix detachment. Okay, so starting with that first story, again, we didn't know how it was that cells died. We didn't know that if cells were matrix detached and they lack and they don't die by anoecus, they're dying by some sort of a caspase independent death. So how is that happening? And one of the things we started thinking about was if there was a role for programmed necrosis in eliminating ECM detached cells. So I think this finding, the idea that other types of cell death, that apoptosis is not the only type of programmed cell death, that finding has been a number of years in the making now. It still feels fairly recent because I came about in an age where, by definition, programmed cell death meant apoptosis and everything else was accidental or as a result of some sort of physiological change. But it's now clear. Necrosis itself can be genetically programmed. And you'll see the term often necroptosis, which is just sort of a play on words between necrosis and apoptosis as a term to define programmed cell death by necrosis. And it often involves the activation of a family of kinases known as the RIP kinases. And they're sort of circled in this pathway here on the left. So we started with a really simple and straightforward question. Is RIP kinase activation responsible for cell death, or at least contributing to cell death during matrix detachment? And the work I'm going to show you here was presented by one of my former graduate students, Dr. Mark Hawk, and he really was sort of the leader on this project. Okay, so simple question. Does ECM detachment serve as a signal to activate RIP kinase 1? I've got a couple of the players in the pathway just for your reference, shown here on the right, as well as many arrows, because I did promise on Twitter that I would try to make my talk with as many arrows as possible, because I think we need to teach our kids the importance of using arrows correctly in talks, right? This is just to sort of serve as a reference point. We did this experiment in MCF10As expressing BCL2. The reason we use BCL2 overexpression is because we wanted to block anoecus, so we didn't have to contend with any impact from the caspase dependent death. And what you're looking at here is just a Western blot for RIP kinase 1 or phosphorylation of RIP kinase 1 at serine 161. You can see in both cases, you see a strong induction in both total RIP kinase 1 and in the activation as measured by phosphorylation here. So it does look like RIP kinase 1 is activated and induced in response to matrix detached conditions. One of the reasons that is because there's also an induction of a deubiquitinase, CYLD. CYLD is also induced in matrix detached conditions, and as a consequence, we know that it can deubiquitinate RIP kinase 1, which should in theory allow for RIP kinase 1 to be stable. Here is a sort of a proof of principle experiment suggesting that's the case. These are MCF10A cells with two different knockdowns, two different hairpins against CYLD, and you can see here that that strong induction of RIP kinase 1 goes down significantly when you knock down CYLD. So what I've told you then is that there's a relationship between detachment and the activation of RIP kinase 1. But the question is, does it matter? You could have an activation of RIP kinase 1 that may not be relevant for whether these cells live or die. And so the first experiment Mark did was to look at a kinase inhibitor of RIP kinase 1, necrostatin 1, which you can see here. Necrostatin 1 inhibits RIP1 kinase activity. Here you're using a cell viability assay, you're normalizing with detachment over attachment, and you can see a nice increase in cell viability. This is measured by Alamar Blue, which is really a measure for mitochondrial reducing potential. And I'll get into why that's relevant a little bit later on. So it does look like treatment with a RIP kinase inhibitor does improve cell viability. It's one of these things where it does not actually have a major impact on cells when they're attached. It only seems to improve viability in matrix detached cells. Here is another way to look at that same question. This is going back to that three-dimensional cell culture system that I mentioned before. So we're looking here at MCF10A cells grown in matrigel, and they form these acinar structures. Here in this pictorial schematic here, you're looking at images taken by confocal microscopy, and we've scored our structures as either clear, mostly filled, or filled. This is just kind of giving you an idea of what those structures would look like in those categories. Basement membrane is marked, or extracellular matrix is marked by the presence of laminin. And you can see, if you focus first here on the left, that MCF10As treated just with DMSO, the majority of the structures at this particular time point score as either clear or mostly clear. If you treat with NEC1, you start to see structures show up that are either filled or mostly filled, and you start to see the number that are completely clear goes down substantially. You do the same experiment in MCF10As that are overexpressing BCL2. So again, this prevents the induction of anoecus. It blocks caspase dependent death. And you ask, well, what happens with NEC1 treatment? The time scale is very different. This is at a much later time point than what I've shown you here on the left. But here with DMSO treatment, at this time, the majority of the structures are either clear or mostly clear. If you treat with NEC1, you now have a number of structures that are filled or mostly filled and very few that are totally clear. So this again gives us additional evidence that inhibiting RIP kinase activity can impact the viability of cells that are grown that are lacking proper matrix attachment. How about if we do this in cancer cell lines? So everything I've shown you so far has been in MCF10As. This is a variety of different cancer cell lines. They're all very different, and we picked very different cells intentionally so as to see if this was this impact of RIP kinase 1 and detachment was quite broad. Here you're looking at a HER2 positive breast cancer line, an ER positive breast cancer line, MCF7s, this is another ER positive breast cancer line, and then we've even got HeLa cells, HCTs, which are our colon line, and H460s, which are a lung cancer line. And in all of these cases, NEC1 treatment gives you an improvement in viability, and this improvement is only seen in matrix detached cells. Matrix attached cells do not benefit whatsoever from treatment with NEC1. And again, this we'll come back to using the H460s a little bit more later on, but again, this tells you that RIP kinase 1 may have some sort of a broad capacity to compromise the viability of cells when they are in matrix detached conditions. We did this with NEC1. We did similar experiments with RIP kinase 1 knockdowns just to rule out any concerns about off-target effects with NEC1, and we see basically exactly the same thing. So what's downstream, right? What's downstream of RIP kinase 1? I've shown you that RIP kinase 1 can give you, the activity can compromise viability. If you think about necroptosis, the next logical player to look at is RIP kinase 3. And the next one downstream of RIP kinase 3 is MLKL. And what's interesting is, and I'm not going to show you this here, but this is as close to canon when we think about program necrosis, that RIP kinase 1 and RIP kinase 3 leads to the activation of MLKL, and it's MLKL that can compromise the integrity of the plasma membrane and cause cells to die by necrosis. We did loss of function experiments with both RIP kinase 3 and MLKL, and neither one of them is relevant for changes that we see in matrix detachment as far as changes that could impact viability. So totally dispensable. We can do loss of function with RIP kinase 3, loss of function with MLKL. In both cases, you see no impact on whether cells that are matrix detached live or die. And what that tells us is that there has to be something else that's downstream of RIP kinase 1 that ultimately could be controlling the viability of matrix detached cells. So at this point, we thought perhaps there was some type of matrix, some type of RIP1 dependent, but RIP3 and MLKL independent type of necrosis that we were studying. We got interested in a downstream, in a molecule or a protein called PGAM5, which is a mitochondrial phosphatase. There's been a few studies that have suggested it can operate in different contexts downstream of RIP kinase 1. So we wanted to look at the impact of interfering with PGAM5 downstream of RIP kinase 1. Again, did simple loss of function experiments. First, I'll show you, we wanted to just check the abundance of PGAM5 and to see whether it was altered by matrix detachment. Doesn't appear to be the case. There's robust PGAM5 in all of our cell lines tested here, independent of whether those cells are attached or detached. But when we do the detached experiments, here you're looking at MCF, or when we do the loss of function experiments, here you're looking at MCF10As, Here you're looking at a knockdown for PGM5, 2 different hairpins. This is just proof that the hairpins that the knockdown worked. You see a huge difference in the viability of these cells. So here are the 2 different hairpins. Again, you're looking at viability on the y-axis, and you can see that that knockdown of PGM5 gives you a huge bump in viability. Again, this is detachment over attachment, and basically if you separate those 2 things out, what's interesting is attached cells are not impacted at all by PGM5 knockdown. It's only matrix detached cells that are impacted by PGM5. Now, we know that knockdown of PGM5 can recapitulate what we see when we inhibit RIP kinase 1, but we don't really know at this point whether PGM5 is downstream or not. And so what we did here, Mark did a really clever experiment where, again, he did this with H460 cells. He engineered them to be deficient in PGM5. You can see that here. And you can see that when you overexpress RIP kinase 1 in attached cells, much like you would see happen during detachment, where detachment serves as a signal to induce RIP kinase 1, you can lower the viability of the H460s simply by overexpressing RIP kinase 1. That's shown here on the left. If you do the same experiment, but you do it when PGM5 has been knocked out, then you can see that actually the ability of RIP kinase 1 to drive down the viability is now completely lost. And what that tells you is that PGM5 is absolutely required for the capacity of RIP kinase 1 to lower the viability of cells. So that we felt pretty good about that, therefore PGM5 functioning downstream of RIP kinase 1. So what is PGM5 actually doing? Again, we started off thinking about this as regulated necrosis, but we haven't really come any closer to understanding what's actually happening. We know RIP kinase 1 is an important player. We know PGM5 is an important player, but we still don't actually know what's happening to our cells. We did these RIP kinase 1, PGM5 loss of viability, these loss of function experiments, and we could see that RIP kinase 1, PGM5 could mediate viability changes in a variety of different cells. So it does hold across a lot of different cell lines, but it still doesn't answer that more fundamental question, what's going on downstream of PGM5? And again, PGM5 is localized to the mitochondria. And it is known to be a mitochondrial phosphatase. So is there a role for PGM5 in altering the mitochondria? We found some really interesting papers suggesting that PGM5 is linked to mitophagy, or the selective autophagy of mitochondria, and that this occurs through regulation of PINK1. So we thought that might be interesting to look at, particularly since we haven't looked at the mitochondrial load in matrix attached and detached cells. Not to mention, a lot of our viability experiments were really measuring the mitochondria and their reducing potential anyways. So what we did is we looked first at PINK1, and just to look at, is there a difference in PINK1 in attached or detached cells? And indeed, in matrix detached cells, you can see a significant induction in the PINK1 protein. The other thing that was interesting is if you take matrix detached cells and you treat them with the RIP kinase 1 inhibitor, that's NEC1 shown here, you start to see this cleavage product showed up. And this cleavage product is similar in size to a band that suggests proteolytic cleavage of PINK1, and that proteolytic cleavage of PINK1 can compromise its activity. So it almost seemed like PINK1 activity was high in detached conditions, unless you inhibit RIP kinase 1, in which case that PINK1 starts to be cleaved. In order to validate that, here you're looking at the activity of PINK1 as measured by phosphorylation of ubiquitin. You can see phosphoubiquitin levels are high in detachment, as shown on the left. And on the right here, when we treat with NEC1, that activity goes down substantially. So it basically leaves us with a model where if you inhibit RIP kinase 1 or PGM5, what in matrix detached cells, you get proteolytic cleavage of PINK1, and that proteolytic cleavage can lead to a loss of PINK1 activity. Okay, so again, the fact that PINK1 is involved here seems to suggest a role for mitophagy, but we haven't actually looked at, to this point, whether or not there is a difference in the abundance of the mitochondria in matrix detached cells. And if there is that difference in abundance, is it controlled by RIP kinase 1 signaling? Here you're looking at the abundance of the mitochondrial outer membrane protein TOM20. You can see, and we're sort of using it as, right here we're using it as a surrogate for the abundance or the number of mitochondria. You can see that the TOM20 levels go down dramatically in detachment, but if you treat with NEC1, which is the RIP kinase 1 inhibitor, then TOM20 levels are rescued. So it does seem like there's a relationship between RIP kinase 1 and the mitochondrial abundance. Same experiment here, except instead of NEC1, we're doing shRNAs against RIP kinase 1 or PGM5. Again, detachment, it goes down significantly. Nice rescue when you inhibit RIP kinase 1 or PGM5. So both of those look pretty good. Similar experiment here, except now we're looking at the abundance of MTCO2, which is actually encoded by the mitochondrial DNA, goes down dramatically in detachment. And there is, in this case, it's more of a partial rescue of MTCO2 when you inhibit RIP kinase 1. So it does seem like there's a relationship between RIP kinase signaling and the mitochondrial abundance. We thought it might be a more straightforward way would be to just actually do this by an imaging approach. And this is just, you're looking at a mitotracker stain. Quantification here shown on the right. You start here in the bottom left corner. Red is mitotracker, blue is DAPI. You can see that red stain. If you compare attached here to detached just above it, you can see that red signal starts to go down. But if you do the same experiment in either the absence of RIP kinase 1 or the absence of PGM5, you can see that there is a nice maintenance of that mitochondrial signal in both cases. So that seems to corroborate what we were thinking, that there is that the number of mitochondria is compromised in detachment, and part of the reason for that is the activation of RIP kinase 1. But how do we know, again, the fact that PINK1 is involved suggests that there's something like mitophagy that's being induced, but we haven't really proven that that diminished mitochondrial signal is a consequence of mitophagy. And so what we did here is we engineered our cells to be deficient in the capacity to undergo mitophagy. So start here on the left. This is just an MCF10A cells. Mitotracker again in red compared to DAPI in blue. And you can see that if you look at the fluorescence in red, it goes down significantly in detachment, and you get nice protection with NEC1. Here's quantification of that on here on the bottom. You do that same experiment, but you engineer these cells by CRISPR-Cas9 to be deficient deficient in ATG5. The capacity of detachment to cause a decrease in mitochondria is almost entirely lost. And on top of that, the ability of NEC1, again, which inhibits RIP kinase 1, the ability of NEC1 to rescue that phenotype is also lost. So this again suggests that the autophagy machinery is necessary in order to see this fundamental change in the mitochondria. If you add back ATG5, this is an HA tag version that we've re-expressed, you can see you reestablish that loss of mitochondrial stain and detachment, and you can once again maintain protection by NEC1. So altogether, we felt pretty good about this, that there seems to be a direct relationship between the capacity of RIP kinase 1 to cause lower numbers of mitochondria and the fact that that's likely occurring by mitophagy, in the sense that the machinery that execute mitophagy are entirely required for that change to occur. But this takes us back to something, again, very fundamental. We've shown pretty clearly RIP kinase 1, PGM5, PINK1, induction of mitophagy. This is all associated with changes in cell viability, but what is it about mitophagy that actually compromises the viability of cells in matrix attachment? And we were puzzled by this for quite some time. Again, going back to some of our earlier studies, we knew that reactive oxygen species could eliminate matrix attached cells. And we were interested in whether the induction of mitophagy could alter reactive oxygen species abundance from the mitochondria. And we found something really quite interesting. So if you look here, you're using, in red, we're measuring mitochondrial ROS as measured by using a stain called MitoSox. You can see if you compare attachment here to detachment here, that signal goes up dramatically, but that when you get rid of RIP kinase 1 or PGM5, you see very little mitochondrial ROS. And again, this suggests, what this tells you is that ROS levels are potentially going up in detachment, and that if you get rid of the machinery that executes mitophagy, those ROS levels go back down. Here's a very similar experiment using the mitophagy or using the autophagy machinery. Here again is mitosoc staining. You can see attached versus detached. The red stain, it goes up significantly. That's blocked by NEC1 treatment. You do that same experiment in the presence of ATG5 knockout. What you can see here is that your capacity to induce mitochondrial ROS goes down, is completely lost, and NEC1 now has no effect, mainly because there's no ROS to begin with. And if you re-express ATG5, you can restore this, right? So you have higher levels of reactive oxygen species, and that's again lost by, that's lost when you treat with NEC1. Now, this is something that's really, we still don't fully understand. Mitophagy, as myself and I think many others have always understood it, exists to eliminate damaged mitochondria, which are often producing ROS. But our data suggest here, at least in this context, that mitochondrial ROS are a consequence of the induction of mitophagy, at least in matrix-detached cells. The precise reason why this is the case, why mitophagy is actually causing an increase in ROS, is something we're working on now. And we're interested in, we have a project where we're interested in the capacity of iron in the mitochondria to be liberated from the mitochondria during the mitophagy process, where it could participate in the Fenton reaction and possibly lead to elevated ROS. That's one of the hypotheses that we're currently exploring. So let me just finish up this, this first part of the talk. How is it that RIP kinase 1, if you go back to the whole premise of the study, the idea is that matrix attached cells have to shut off anoecus, and they have to compromise, rewire their metabolism or deal with elevated oxidative stress in order to survive, and that this could have implications for tumor progression. So we did sort of a proof of principle experiment here to look at if RIP kinase dependent activation of mitophagy could potentially impact tumor formation in vivo. So what Mark did, engineered H460 cells, these are lung cancer cells, to be defective in RIP kinase 1 or PGAM5, and then injected them into the tail vein of immunocompromised mice. Here you're looking at a longitudinal assessment of healthy lung volume. So that healthy lung volume goes down as the metastases start to accumulate on the lungs. So blue is PBS, you see no change. Green is just your control cells that over time, as we took this at 2, 4, and 6 weeks, as the lungs start to accumulate more tumor, that the healthy lung volume goes down. But if you look at what happens when you knock down RIP kinase 1 or PGAM5, the situation is even worse. You have more metastasis, more tumor burden, as evidenced by this decrease in healthy lung volume, suggesting that the RIP kinase pathway we've been studying is potentially acting in a tumor suppressive capacity to eliminate incipient metastatic cells that are matrix detached. Another way to look at it here, this is ex vivo analysis. So you're looking at images of the lungs shown down here on the bottom. Here's our control H460s. Here's knockdown with RIP kinase 1 or PGAM5. And you can see if you just measure the tumor burden on the lungs as a %, you can see that there's a significant difference. There is tumor burden in the control H460s, but that when you knock down RIP kinase 1 or PGAM5, there's substantially elevated tumor burden. Okay, so that leaves us with a model that looks like this. And I'll just have you focus on the left for now, that in matrix detached cells, there is an activation of RIP kinase 1. It seems to be regulated by CYLD to give you a increased stability of RIP kinase 1. RIP kinase 1 activity can then cause a complex that forms. I didn't show you the IP data on this, but RIP kinase 1 will physically interact with PGAM5 and PINK1. That stabilizes PINK1, protects it from proteolytic cleavage, and causes mitophagy. And through a mechanism we don't fully yet understand, can lead to an elevation in mitochondrial ROS that can kill these cells. Now, we have lots of questions that we're still pursuing, things that we don't understand. I think probably the most significant is how is it that mitophagy leads to the elevation in mitochondrial ROS, and that's something that one of the grad students in the lab is working on quite hard right now. We started this project looking to try to understand how cells ultimately die, but we didn't actually get there. We don't know how cells ultimately die, other than that this is another way that cells can develop elevated oxidative stress that can kill them in matrix detached conditions. So we're still trying to understand the death mechanism itself. And again, we have preliminary data here suggesting that RIP kinase 1 dependent mitophagy is antagonizing tumor progression, but this obviously requires additional analysis. One thing that we've become quite interested in is a potential relationship between RIP kinase 1 mediated mitophagy and resistance to therapy, where you might think that cells that are resistant to certain cancer cells that are resistant to certain types of therapies may be surviving in part because of shutting off this RIP kinase 1 dependent pathway. And we do have some interesting data coming in suggesting that cells that are resistant to immune checkpoint inhibitors are potentially resistant, have defective RIP kinase 1 mitophagy in detachment. Okay, so I think what I'll do is I'll go on to the second part of this talk, shifting gears a little bit and focusing here now on what are the signaling pathways that can actually rewire metabolism during matrix detachment in cancer cells. And this work really follows, this work follows from a paper that we published a few years back that suggested that this kinase SGK1, serum and glucocorticoid kinase 1, could operate downstream of RAS mutations to promote ATP generation and in addition to promote survival during matrix detachment. This is a schematic from that paper, and here's the citation if you're interested. But one of the things that's interesting, SGK1 is an AGC family kinase, much like AKT. It's an effector kinase of PI3 kinase, and yet it seems like it may have an important role, at least in this context, in promoting ATP generation. Now, again, it still left us with some interesting questions, which is, we knew that this was important downstream of RAS, but we didn't know if SGK1 itself, if activation of SGK1 was sufficient to give you changes in metabolism that would promote survival during matrix detachment. And we didn't really understand the mechanism by which SGK1 could promote the enhanced ATP production. And so that's really what I'm going to show you here today. That work has been led by former graduate students in the lab, Dr. Josh Mason, shown here, and current graduate student Jordan Cockfield, shown here on the right. Jordan actually just passed her oral qualifying exams just last Friday on Zoom, which is the first time that we've attempted to do any sort of exam, any sort of formal exam on Zoom, and she did really well. So again, basic question: is it enough? Is SGK1 activation sufficient to promote ATP generation when cells are grown in matrix detached conditions? For these experiments, we wanted to compare activation of SGK1 to activation of AKT. So what you're looking at here, if you start on the left, we're measuring ATP levels in detached cells. This is using a mouse breast cancer line, 4T07s, and empty vector here. The S422D mutation is a constitutively active version of SGK. And then we're using a constitutively active version of AKT. It's myristylated version that locks it to the plasma membrane and renders it constitutively active. And you can see only SGK in detached cells can really drive up ATP levels quite significantly. You do the same experiment in attached cells, and you do see some capacity. You see some changes perhaps, but not nearly, nothing that would be statistically significant. And importantly, you don't see any differences between the S422D, which is the SGK activation, and the myristylated AKT. Now, here you're just looking at confirmation of our engineering of our cell lines. So here in 4T07s, you can see phospho-NDRG. This is a downstream target of SGK. You can see that our cells expressing the S422D give you elevated phosphorylation. That's what we would expect. This is just total SGK1, again, what you would expect. And here is confirmation of AKT activation as measured by phosphorylation of AKT. Here you're looking at phospho-GSK3 beta, and you're looking specifically at a phosphorylation site that is hit by both SGK and AKT. So if these results up here on the left were because SGK1 just happened to be, we happen to have higher levels of SGK activity in our S422D when compared to this myristoylated AKT, then you would expect that to show up here on a site that is phosphorylated by both of these kinases. And that's not what you see. You see equivalent phosphorylation of GSK3 beta in both the S422D and the myristoylated AKT conditions. So what that tells you is there's something specific about SGK and something specific about matrix detached cells that allows SGK1 to promote ATP generation. Okay, so we did this in other cell lines. Here you're looking at a human triple negative breast cancer line, MDA-MB468. Significant change in ATP levels with S422D, not so with myristoylated AKT. And shown here on the right, when you look in matrix attached conditions, you see no difference between any of the conditions. Similar analysis here. These three blots are telling you confirmation that we've engineered our cells correctly. And here on the bottom, you can see again measurement of phosphorylation at that one GSK3 site. And if anything, you actually have higher activity of AKT as measured by this particular phosphorylation. And again, despite that, you still see a specific induction of ATP only when cells are grown in detached conditions. So we flipped this. We knew SGK1 was sufficient, but is it necessary for ATP generation? We had shown that was the case in cells expressing oncogenic RAS, but we were not sure if this held true in broader context. Here you're looking at 4 different cell lines. Again, they're all very different, and we've intentionally selected different cells to look across a broad variety. 4T07s are a mouse breast cancer line. HCTs are a colon cancer line. Human triple negative breast cancer shown here with the 468s, and this is a HER2 positive human inflammatory breast cancer line, the KPL4s. Each of these cells, they were treated with this EMD compound. This is an SGK1 kinase inhibitor. And what you can see here is ATP levels are dramatically compromised in all 4 cases when you inhibit SGK. Again, this is only, and to be clear, this is only happening in matrix detached cells and is not happening in matrix attached cells. What about Anchorage independent growth? This is a nice way to measure long-term survival of cells that are lacking proper integrin mediated matrix attachment. Here, you're looking at first shRNA against SGK1. So that's instead of that EMD treatment. And again, 2 different hairpins in all of these cases. Substantial reduction in ATP levels in detachment. And here you're looking at colony count in a soft agar assay. And similarly, you can see that the number of colonies that form in soft agar experiments goes down really dramatically. So it does suggest that not only does SGK1 promoting ATP generation during detachment, but that is also, that activity is also critical for the long-term survival of those cells in matrix detached conditions, as we would see in these Anchorage independent growth assays. Okay, so again, going back to the mechanism, what is actually happening here? Why is it that SGK1 mediated ATP generation, which is happening during detachment, how is it actually causing an increase in ATP? There is some evidence suggesting a relationship between SGK1 and glucose uptake. We looked there first. Here you're looking at glucose uptake measurements comparing in those 4 cell lines. Again, you can see that there's an elevation in glucose uptake with SGK1. You see it with AKT, although not as dramatically. That's true in all 4 of these different cell lines. And if you look at the converse experiment where you do an shRNA against SGK1, you can see that using 2 different hairpins that the amount of that glucose uptake goes down dramatically. So it does look like there's a direct relationship between SGK1 and glucose uptake. How is it that SGK1 promotes glucose uptake? Well, it turns out if you look at the levels of the GLUT1 protein, SGK1 can very strongly lead to high levels of GLUT1. Here on the left, you can see you're just looking at a Western for GLUT1. SGK1, again, the S422D mutation shows you high levels of GLUT1 in detachment. If you do a knockdown using 2 different hairpins, then that GLUT1 goes down quite dramatically. Here you're looking at, well, what's the source of the increase in GLUT1? It's actually a transcriptional change. It's a transcriptional change in GLUT1, where you can see here's your baseline. Here is SGK1 activity in the presence of the DMSO, which is your control for your inhibitor. And then when you treat with the SGK1, if you treat with the SGK inhibitor, then that GLUT1 expression as measured by mRNA goes down. Here you're looking at the relationship between the glucose uptake and GLUT1 itself using this compound, which is a GLUT1 inhibitor. Again, increasing glucose uptake with SGK1, you inhibit GLUT1 and that increase in glucose uptake goes away. So we're left with, again, a model where SGK1 is promoting GLUT1 transcription. We don't yet know the identity of the transcription factor or transcription factors that are promoting GLUT1 transcription. And as a consequence of that elevated GLUT1, protein, you get increased glucose uptake, which is responsible for the increase in ATP generation. Okay, so now that these cells are taking up glucose at high levels, is that it? Is it just that glucose uptake is coming in at high levels and that's responsible for the elevated ATP generation? We were interested in assessing the contribution of different components of ATP generation. So we first wanted to look at the contribution of the mitochondria. We figured that again, the major source of ATP in most respiring cells is going to be the TCA cycle. And so we wanted to look at and see if the consequence of the elevated glucose uptake was elevated mitochondrial production of ATP. Here is an experiment where on top you're looking at, we did an inhibition with SGK1, the SGK inhibitor. This proves that the inhibitor works. And then we tried to rescue the ATP levels by simply feeding the cells cell-permeable methyl pyruvate. So if these cells are defective in glucose uptake but maintain functional TCA activity, the idea would be you could simply feed them cell-permeable pyruvate and you could rescue the ATP levels if that was the source of your elevated ATP. But that's not the case. You treat with different doses of methyl pyruvate here in the presence of EMD, and that does not rescue your ATP levels in these cells. We did this experiment, we sort of looked at this a different way. Here we used a mitochondrial uncoupler, CCCP. If you look here, let's start actually here on the right in the empty vector controls. If you look at ATP levels, you either treat with 2-deoxyglucose, which interferes with glucose metabolism more broadly, or CCCP, you can see a dramatic reduction in ATP levels. But what's interesting is you take those same cells, you overexpress the constitutively active version of SGK, and now the ability that ATP levels, those ATP levels that you're getting, are insensitive to CCCP treatment. So it really suggests that it's not the mitochondria. The mitochondria are actually, the activity of the mitochondria, the activity of the TCA cycle is actually dispensable for that elevated ATP, those elevated ATP levels you see with SGK1. So we wanted to probe this a little bit further and look at if the TCA cycle function is it in fact dispensable for SGK1 mediated ATP production. And we collaborated with Nav Chandel at Northwestern to sort of address this question. Nav's lab has generated these really nice cells that are really helpful for studying cells that lack TCA cycle function, or at least oxidative TCA cycle function, while simultaneously maintaining mitochondrial membrane. Basically, the way these have been set up, these cells, they have a doxycycline inducible dominant negative mutation of DNA polygamma that's shown here. And they've also been genetically engineered to eliminate ATPIF1. And so what happens is when you give these cells doxycycline, you deplete the mitochondrial transcripts. And when you eliminate ATPIF1, which is an inhibitor of the F0, F1 ATPase, then you can restore the mitochondrial membrane potential in these cells. So you can have cells with potential, but they have defective oxidative TCA flux. We took these cells, we got these cells from Nav, engineered them to overexpress either constitutive activation of SGK1 or of AKT, and then similarly grew these cells in matrix detached conditions to see what would happen when you completely deplete mitochondrial, when you completely disable oxidative TCA cycle function. Here on the right, you can see doxin administration gives you a complete loss of MT COX2. So that's shown here. That tells you that we've been successful in actually depleting these mitochondrial transcripts. And then here you ask, well, what happens now when you put in SGK? And actually what happens is exactly the same. SGK1 can still drive high levels of ATP production. So that tells you again that it does not seem to be that whatever SGK1 is doing to promote ATP generation is not a consequence of, is not dependent on oxidative TCA cycle function. So we were then interested in what's the fate of glucose once it comes into these cells. What is the fate of glucose-derived carbons after when you get SGK1 mediated glucose uptake? And for this, we collaborated with Jason Lopesal at Duke, and we did some glucose tracing experiments where we used glucose that's been labeled at the 1,2 position, and you can see intermediates that are labeled M+2 are going to be largely in glycolysis, and intermediates that are labeled M+1 as a consequence of this decarboxylation here are going to largely label as M+1. And it was interesting that, as expected, we see a huge increase in all of these different glycolytic intermediates shown here on the left and labeling M+2, but we also see a huge increase in the pentose phosphate pathway intermediates, which are labeled M+1. So it was that PPP flux that was really interesting to us, because we had seen previously that the pentose phosphate pathway could be particularly important in matrix detached cells. So we asked whether the PPP itself could contribute to SGK1 mediated ATP generation. Here we're using an inhibitor of the pentose phosphate pathway flux, 6AN, and indeed, if you interfere with the pentose phosphate flux, then ATP levels go down substantially. This is in 4T07 cells expressing constitutively active SGK. You do a similar experiment, but instead of using an inhibitor, you do a knockdown of glucose 6-phosphate dehydrogenase. Here's confirmation of the knockdown on the right, and again, you see a significant decrease in the ATP levels. When you do when we do these Anchorage independent growth assays, we looked in soft agar. Colony count measured here, 6AN treatment causes colony count to drastically be reduced. And so this is again suggesting that the PPP is particularly important in matrix detached cells that have activation of SGK. And we were interested, well, what we'd shown previously is that the PPP derived production of NADPH could lead to lower levels of reactive oxygen species. So we thought maybe if we could provide the NADPH through an alternative source, in this case, the addition of methyl malate, which is a cell-permeable version of malate that can generate NADPH through a completely separate pathway through malic enzyme, then that might be enough to rescue the ATP levels after we inhibit the PPP. But it turns out that that's not the case. So 6AN treatment shows here, it does give you a significant decrease in ATP, but malate treatment at actually quite high concentrations does not rescue. So again, we've got a role for PPP, but we don't know precisely why the PPP is so important for ATP generation in cells expressing SGK1. And we're left with this question, how is that actually happening? And we started thinking about another model where, again, we have very high levels of glucose that are coming into these matrix detached cells that express SGK1. And is it possible that potentially these cells are largely glycolytic, that they're using glycolysis to make ATP, but what's happening is the pentose phosphate pathway can accommodate a lot of that carbon flux and then redirect it back into glycolysis. And it turns out the way this can happen, you look at this diagram here, is through the production of glyceraldehyde 3-phosphate, or G3P. This can be produced in the pentose phosphate pathway and then reincorporated back into glycolysis for ATP generation. So what we did is we used an activator of GAPDH, which is glyceraldehyde 3-phosphate dehydrogenase. This is the enzyme that would metabolize G3P in glycolysis. We used an activator of GAPDH on this compound NMN and asked, could we rescue ATP levels when you've inhibited the pentose phosphate pathway? And the answer here is actually yes. You activate GAPDH with NMN, and now you can restore the high levels of ATP that you see in cells that where it was lost with the inhibition of the pentose phosphate pathway. You can do the same experiment here. This is, instead of using 6-AN, the pharmacological approach, we do a genetic knockdown of glucose 6-phosphate dehydrogenase. And then when you treat with NMN, you get a significant elevation, again, in ATP levels. So again, suggesting that that GAPDH activity, which metabolizes G3P, can, if you stimulate that activity, then you can rescue ATP generation. That really, that pathway, that part of that point in the pathway is critical for ATP generation in these SGK1 expressing cells. And here what we did is, rather than activate GAPDH, we tried to add back glyceraldehyde 3-phosphate, or G3P itself. G3P does not cross the plasma membrane. So you have to permeabilize the membrane with low doses of detergent. We use 0.01% saponin here. So if you look here, you're looking at 4TO7s. They lose ATP levels when you treat with 6-AN. G3P does not rescue here because we haven't permeabilized the membrane. But when we permeabilize the membrane here on the left, then ATP levels start to go back up. Again, telling you that G3P levels from the pentose phosphate pathway seem to be critical for driving ATP levels in the matrix detached cells. Similar experiment here, except instead of inhibiting the pentose phosphate pathway with 6-AN, we're using an SGK kinase inhibitor with this EMD compound right here. And again, measuring ATP, you start on the right. You can see EMD causes a decrease in ATP levels. G3P does not rescue, again, because the membrane is not permeabilized. You do the same thing here in the presence of 0.01% saponin, and you see a significant rescue in ATP levels. And then here, you're looking at, instead of EMD, we did this with our knockdowns with SGK1. Again, starting on the right, this is the non-permeabilized membrane. Knockdown of SGK gives you a significant loss in ATP. You can't do anything with G3P to rescue it. Here, when you permeabilize the membrane, you can now get G3P into these cells and rescue ATP levels. So this was an experiment. I remember when Josh brought this to me. Josh, if you're watching, you'll remember this as well. Josh brought this experiment to me, and I told him there's no way this was going to work. And this is one of the many cases where I'm proud to have been proven wrong by my own grad students. Okay, so in summary, activation of SGK1 in matrix detachment leads to an elevation of GLUT1 protein. That elevation is transcriptional in nature. GLUT1 protein causes elevated glucose uptake in these cells, and that glucose uptake is critical for ATP generation, but it's also critical, but a major part of that is accommodating that carbon flux in the pentose phosphate pathway, and that you need G3P production from the pentose phosphate pathway to continually stimulate, provide a substrate for GAPDH to generate ATP levels. This second part is entirely unpublished. We literally just published a preprint of this manuscript. Here's the link for it. I'll put the link up on Twitter as well in case anybody's interested. I'm new to the preprint game, so I'd love to have any feedback, just to get any feedback from people that were out there about thoughts on our findings. Okay, so I will close here. Lots of people to thank. I'm really privileged to work with a number of great students. A couple people I'll highlight. This is my current graduate students and postdoc. I highlighted the work of Jordan Cockfield on the SGK1 story. Former lab members, Mark Hawk, former graduate student, led the RIP kinase story. Josh Mason, in addition to Jordan, were sort of the leaders of the SGK story that I presented in the second part of the talk. We've got a phenomenal group of undergraduate students here at Notre Dame that contribute meaningfully to the lab. These are the current undergrads in the lab. We're missing them all right now. They're all stuck at home with COVID-related quarantines. So hoping that they're all doing well, but we really have a great group there. And then a number of great collaborators. I didn't mention all of these people. UT Southwestern, Ralph DeBerdinis and Lei Zhang helped us with some IDH2 related experiments in the RIP kinase story. I didn't even get a chance to talk about that data. Mike Overholzer at Sloan Kettering and Jens and Monica from his lab helped us to really narrow in on studying RIP kinase mediated mitophagy and confirming that it was in fact a mitophagy response. I mentioned Nav Chandel at Northwestern with the 293 T cells that are deficient in the oxidative TCA cycle function, and INMA from his lab. Also great collaborators. And then at Duke, Jason Locosal and then Wan Lu and Xiaojing Lu helped us with the carbon tracing experiment. So I'll end there. And again, I just want to thanks to Phil, the White McGarrett Lab for this initiative. It's been, again, there's not a lot of normalcy right now. It's nice to have some, and really just awesome I was thrilled to be able to participate in this. And thanks to all for all the speakers and everybody participating. It's just nice to hear about exciting science while we're all cooped up here in the house. So hope everybody's staying healthy and safe. And thanks again. And I'm looking forward to participating in the rest of the series.